Hello students, Namaskar. Today we will be studying about the reproduction in organisms. This is the first chapter in class 12th biology, and if you compare it with class 11th, then this is the 6th unit, reproduction. This unit consists of 4 chapters. So today we will be starting with the first chapter, that is reproduction in organisms. You all know that reproduction is a characteristic of all living things. All living things reproduce. Every living thing that once born on the face of earth grows, reproduces, and then dies. Now, this span of life, or this period of life of an organism between birth and final death of the organism is called as the lifespan. The lifespan of any organism can be divided into basically 4 phases, starting off naturally from birth to the juvenile phase, or the phase in which the organism grows and attains maturity, reaching to the second phase, that is maturity, which is also known as the reproductive phase of an organism. It is in this phase that the organism is now mature enough to reproduce organisms of its own kind. Then the next phase is aging, or we also term it as senescence. After the organism has reproduced, it undergoes further deterioration and aging, which finally leads to the death of the organism. In this chapter, we will be discussing about the reproductive phase. Reproduction is important because, one, it ensures continuity of species on the face of earth. Secondly, it also helps in bringing about variations. Now, what are these variations? How they are produced, and what is its significance? Variations are the factors, or there are certain changes in an organism that take place for the organisms to adapt to the changing environmental conditions. So, variations are very important, and reproduction is a process which ensures variations in organisms. Now, how it exactly happens, we'll be dealing with that in our further chapters. Third importance of reproduction is the organization of organism, the population organization. Rather, we can say that it ensures the population organization. Now, what is population organization? It is with the help of reproduction that a particular population can be divided into the young, the older, the reproductive age groups, and so on. Above all, the main importance of reproduction is that it ensures life on face of earth. So, today we'll be discussing in detail the different types of reproduction. Before we move further, let us discuss about lifespans a little more. What are these lifespans? As we said, lifespan is the duration of an organism's life between birth to death. Let's consider the lifespan of some organisms. For example, the lifespan of an elephant is about 60 to 90 years. That of a mayfly can be as small as 1 day. On the other hand, a tortoise can have a lifespan of about 200 years. Among the birds also, there is a lot of variation in the lifespan. A crow has a lifespan of about 15 years, whereas another bird, parrot, has a lifespan of about 140 years. So you can see here that the lifespan of an individual is not dependent on the size of the individual. An elephant, which is so large, lives for only 60 to 90 years, whereas a parrot for 140 years. On the other hand, parrot and a crow have a similar size and they both are birds, but still you can see there's a lot of variation in the lifespan in both of these individuals. The plants have a longer lifespan as compared to animals. Like, you can take the example of banyan tree. It has a lifespan of about 2,000 to 3,000 years. And the redwood tree, still higher, 3,000 to 4,000 years. So we can see that the lifespan in an individual varies. Now, after this, let's take up the different types of reproduction. The reproduction can be basically divided into two types: the asexual reproduction and the sexual reproduction. Let us do a comparison of these two types before we move further to discuss about them in detail. Asexual mode of reproduction is a kind of reproduction in which only one parent is involved. Or we can say it is uniparental. In sexual reproduction, always two parents are involved. They are biparental. In asexual reproduction, gametes are not formed, whereas in sexual reproduction, gametes are formed. In asexual reproduction, only mitosis or simple cell division takes place, whereas in sexual reproduction, both mitosis as well as meiosis take place. Since there are no gametes formed, so there is no fertilization in case of asexual reproduction. Whereas in sexual reproduction, fertilization takes place. Then, asexual mode of reproduction is usually found in the lower invertebrates as well as in lower chordates. Whereas sexual mode of reproduction is found in all animals and most of the plants. So, all animals and most of the plants. Now, since only one parent is involved and no fertilization is taking place, the daughter cells that are formed, or the daughter individuals that are formed, they are genetically similar to the parents. Whereas in case of the sexual reproduction, variation takes place as there is a combination of gametes between when sexual reproduction is taking place. So the individuals, the daughters that are formed are not genetically similar to parents. So these are the basic differences between the asexual and sexual mode of reproduction. Now, in the next part, we'll be discussing about the various modes of asexual reproduction, how they occur in different organisms, how they help in continuity of the species. So all these things will be taking up in detail one by one. So if we look, we have already covered two things here, the lifespan, and we have covered the types of reproduction. Now we will be taking up the first type, that is fission. Fission is a kind of asexual reproduction which is similar to simple cell division. This can be seen in various protistans, some monerans, and some simple alga. Now, fission can be basically divided into three types: binary fission, multiple fission, and plasmotomy. Let us take these types one by one. Binary fission. This is a kind of asexual reproduction in which, as we started off, only one parent is involved. So what happens that in organisms such as amoeba, when they grow in size, their size increases, they divide to give rise to two daughter cells by simple cell division. That means first the nucleus divides into two, followed by the division of cytoplasm into two daughter cells. These daughter cells, when increase in size, they further divide to give rise to two new daughter cells. You can see in this kind of division, no biological death of the parent is taking place. Hence, these organisms are said to be immortal because one parent dividing to two daughter cells, and this further continues. So binary fission can be seen in protistans like amoeba, euglena, paramecium, and so on. Next we come to multiple fission. Now, what happens in multiple fission? Multiple fission can be seen in amoeba also. During the unfavorable conditions, amoeba develops a cyst wall around itself. So what happens? That it withdraws its pseudopodia and it develops a protective three-layer covering around itself. And when does that happen? It happens in the unfavorable condition. This amoeba, it divides continuously and give rise to many daughter cells. When the conditions become favorable, the outer protective layer of the cyst, it ruptures, and these amoeba, they are later expelled or dispersed into the environment. So if this is the outer covering layer, so after it ruptures, many amoeba, they are dispersed into the environment, and these daughter cells then thrive as single independent individuals. So this gives rise to the many daughter cells. So what is happening here? Multiple cell divisions, and this is called as multiple fission. Multiple fission can also be seen in Plasmodium, which is a protozoa which causes malaria. You must be knowing that the malarial parasite, it completes its life cycle in the female Anopheles mosquito as well as in the human beings. So, during this period, it undergoes multiple fission to give rise to many sporozoites which further grow and cause the infection. Multiple fission, as you can see, takes place or can be seen in two organisms. One is amoeba and the other one is Plasmodium, as we were discussing. Now, the third type of fission is plasmotomy. Now, plasmotomy is seen in multinucleate protozoans like Opalina. So, the example is Opalina. Here, the examples are amoeba and Plasmodium. And here, the examples are amoeba, euglena, and paramecium. So, how is it different from the binary fission and the multiple fission? In plasmotomy, what happens? That organisms like Opalina, they have large number of nuclei. For example, we have an Opalina having 10 nuclei. So, how it will divide? First, one organism will divide into two daughter cells by division of its cytoplasm, that is cytokinesis, and give rise to two daughter cells, each having 5 nuclei. That means these nuclei are equally divided into the two daughter cells. After that, in the next stage, the karyokinesis takes place, that now the number of nuclei are again increased in each daughter cell, and after some time each daughter cell, it develops 10 nuclei each. So we can see here that in binary fission, first karyokinesis takes place, that is division of nucleus takes place, followed by cytokinesis. Whereas in plasmotomy, the cytokinesis first takes place, followed by karyokinesis. So, after the daughter nuclei are formed by division of cytoplasm, the nuclei, they divide amongst each other, and the daughter cells then have 10 nuclei each, and then this process repeats. So, these are the three types of fission, the first mode of asexual reproduction that we have discussed here. So, this is done. Now, we will be coming to the second mode of asexual reproduction, which is called as budding. Budding can be seen in unicellular fungi such as yeast. The buds that are formed are of two types. They can be formed internally, known as internal buds, or they can be formed on the outer body surface. So, we can divide budding into two types: exogenous budding or exogenous buds and endogenous buds. So, depending upon the location at which the buds are formed, it can be of two types: exogenous and endogenous. Exogenous budding is seen in organisms such as yeast. What happens in a yeast cell? The yeast organism, it's a single-celled organism. When its size increases, the nucleus, it begins to divide, and a small protuberance appears on the surface of the yeast cell. So we can see here the nucleus dividing and the bud appearing. After the nucleus has divided into two, the bud, it gets a nucleus of its own. It is still attached to the parent body, and when it becomes heavy, it detaches itself and gives rise to a new individual. So this kind of asexual reproduction is called as budding. External budding or exogenous buds can also be seen in certain coelentrates like the hydra. You must have studied the budding of hydra in your previous classes as well. So what happens in a hydra? A hydra is a sedentary coelentrate. It has the presence of tentacles. So a small bud appears from any of its surface. It grows in size and eventually it breaks off from the parent body, and when it finds a suitable substratum, it grows to give rise to a new individual. So here also we can see that the buds are formed externally on the outer side. What are the endogenous buds and how does that budding take place? So we can write here the exogenous buds are found in yeast and in hydra. Endogenous budding. Endogenous budding is seen in a freshwater sponge also named as spongilla. Spongilla makes these gemmules in the inner surface of its body to tide over the unfavorable conditions. So how exactly these internal buds are formed? Let's see in detail. So as you know, these sponges, they are again fixed to the substratum. They have a porous body. They have pores over their surface. So on the inner surface of their body, so this is the covering. So on the inner surface of their body, they produce gemmules like this. Okay. What do these gemmules look like? They are something like this. Each gemmule is covered by a protective covering. So this is the outer covering of the gemmule and this is the inner covering. And inside are present large number of cells. These cells later on they form a new colony when these gemmules reach a surface where they can lodge themselves and form a new colony. These cells are called as archaeocytes. These cells are known as archaeocytes. And this is a small covering on this this is a small opening called as the micropyle, which helps in dispersing these archaeocytes or the cells which are formed inside the gemmules. Gemmules also have the presence of spicules on its surface, which is protective in nature and protects the gemmule from harsh conditions. So, when these gemmules are liberated from the body, they fall upside down and from here they kind of fix themselves into the substratum and give rise to a new sponge body or a new individual. So budding, we have seen, is of two types. They can be external buds formed or the internal buds can be formed. So budding can be internal as in yeast and hydra, and external as seen in freshwater sponge or spongilla. Okay, so next we come to the third type of asexual mode of reproduction, and that is fragmentation. Fragmentation is a kind of reproduction in which fragments from the parent body, they have the ability to grow and form a new individual. Fragmentation fragmentation can be seen in organisms such as Spirogyra. Spirogyra is a green algae and it is filamentous in nature. It has linear rows of cells. You must have studied about it in class 11 in plant kingdom, and it has got the spiral shaped chloroplast. It divides by fragmentation. One of the modes of its asexual reproduction is fragmentation. What happens that these cells, they divide at any point, and when they reach a suitable substratum, they give rise to a new individual. So they can form new individuals. So if this is a parent body, it gives rise to a new individual, or the daughter cells. It gave rise to the new individual or the daughter cells.